Good morning, everyone. Today we are going to discuss about the third part of bioinorganic chemistry, that is photosynthesis. So, what is photosynthesis? It is actually a chemical reaction. The reactants are carbon dioxide and water, and in presence of sunlight, they produces carbohydrate and oxygen, and this is the reaction. So, it actually taking place in chlorophylls. So chlorophylls act as photosensitizer in photosynthesis and is a complex molecule. The two most common types of chlorophylls are chlorophyll A and chlorophyll B. Chlorophyll has a porphyrin ring system similar to that of hemoglobin. The difference is in chlorophyll the central metal is magnesium and it is in +2 oxidation state. The porphyrin ring is there. The central metal is magnesium. Instead of iron, in chlorophyll you have magnesium, and in hemoglobin there is a histidine unit, and for that we have lipid soluble hydrocarbon tail in chlorophylls. So there is some word similarities with the structures of hemoglobin. The difference is the central metal is magnesium and it is in +2 oxidation state, and instead of histidine you have a lipid soluble hydrocarbon tail in chlorophylls. So chlorophyll A is the main photosynthetic pigment and is directly involved in light reaction of photosynthesis. So light reaction taking place in chlorophyll A, and we know that light reaction taking place in grana. Keep in mind. So all other pigments which include chlorophyll B, xanthophyll and carotenoids are auxiliary pigments. The auxiliary pigments means they do not directly involve in photosynthesis, but they just help the chlorophylls to absorb light and pass energy into chlorophyll B, and this is what other pigments are doing, and it is considered as auxiliary pigments. So they do not directly participate in photosynthesis but are able to absorb light and pass the energy to chlorophyll B. And this is the structure of chlorophyll, and in hemoglobin we have central iron here, but in chlorophyll you have magnesium in +2 oxidation state, and we have some hydrocarbon chains, that's all. Then what is photosynthesis? And it is the overall chemical reaction of photosynthesis may be regarded as the combination of water and carbon dioxide photosensitized by chlorophyll to form carbohydrate and oxygen. The photosynthesis is complex process which can be broken down into two reaction stages: light reaction or light dependent reaction. And we know that light reaction taking place at grana. In plants, light reaction takes place within the chloroplast thylakoids, where the chlorophyll pigments reside. When the light reaches the pigment molecules, it energizes the electron within them, and these electrons are involved in electron transport chain in thylakoid membrane. See, when the chloroplast containing thylakoids exposed to sunlight, the electrons gets energized, and this energy is stored as either ATP or NADPH. Then this ATP or NADPH may be used for the dark reaction as a energy source. Then during the process, high energy chemical intermediates ATP and NADPH are formed. So the electronic excitation energy is stored as ATP. We know that adenosine triphosphate and NADPH are formed. So each chlorophyll replaces its lost electron from water. See, whenever there is a excitation of electron, the electrons are removed or ejected. So these lost electrons are satisfied by decomposing water. So water may be decomposed to hydrogen and oxygen, and during the process the electrons are ejected and those electrons are captured and filled in chlorophyll. And that's how oxygen is produced during photosynthesis. So each chlorophyll replaces its lost electron from water. That is, water is splitting and oxygen formation takes place. So in a light reaction, 12 NADPH and 18 ATP are formed. ATP and NADPH are energy carriers that carry energy to the dark reaction. Keep in mind. So what is dark reaction? It's actually light independent reaction. Dark reaction takes place in stroma. During this process, energy from the ATP and NADPH molecules generated by light reactions drive a chemical pathway that uses the carbon in carbon dioxide to build 3-carbon sugar called glyceraldehyde 3-phosphate, also called G3P. Then cells then uses G3P to build a wide variety of other sugars like glucose and other organic molecules like glucose, fructose, maltose, etc. The products of these reactions are then transported to other parts of the cell where they are broken down to make energy carrier molecule to satisfy the metabolic demands of the cell. In plants, some sugar molecules are stored as sucrose or starch. Basically, glucose is produced, then its subproducts may be developed as per the some mechanisms present in some plants. Now, that's all about the photosynthesis. Now we are going to the very important portion of the module, that is sodium potassium pump. And for your exam point of view, these points are very important. Sodium potassium pump. See, a pump. Here there is a term called pump. What is the role of a pump? It actually helps to carry something. Against energy gradient. So I will give you some examples. Suppose there is some water in a well and I want to bring this water to a tank in second floor. So the potential energy of water inside the well is very low, and I am using a pump to carry this water to the tank present in second floor. And we know that in second floor definitely there will be very high potential energy. So here we are using a pump to transport something against energy gradient from the site of lower energy to a site of higher energy. We are transporting water by using pump. So in our biological system, instead of potential energy, concentration gradient is used. We know that osmosis. In osmosis, the transport takes place from the site of higher concentration of solvent to lower concentration of solvent. Okay, so generally it is a free process, does not require energy. That will takes place by itself. It is a free process that doesn't require energy. Osmosis is a free process, but reverse osmosis, it requires some energy. And this is how a sodium potassium pump works. So it is a pump in our biological system that works against the concentration gradient of sodium and potassium. So let's discuss. In animals, the concentration of K plus in intracellular fluids, means the fluid inside the cell, is about 0.15 molar, and that of Na plus is about 0.01 molar. The concentration of K plus ions in the extracellular fluid is less than 0.004 molar, and that of Na plus is, there is a correction, keep in mind, it's 0.01. Actually it is 0.1. Okay, here it is 0.01, correct it as 0.1. So in extracellular fluid, the concentration of Na plus is higher, Whereas in intracellular fluid, the concentration of K plus ions will be higher. Okay, that's the situation. So inside the cell, potassium ion concentration will be higher. Outside the cell, sodium Na plus sodium ion concentration will be higher. So the sodium potassium pump works against the concentration gradient. Okay, the concentration gradients of these ions are maintained by a mechanism called sodium potassium pump by the active transport of Na plus and K plus ions across the cell membrane. So actually sodium ions are transported from intracellular fluids to extracellular fluids. Similarly, the potassium ions are transported from extracellular fluids to intracellular fluids. Basically, both ions are transported from the site of lower concentration to a site of higher concentration. So, for such as transportation, which means from the site of lower concentration to a site of higher concentration, it requires energy like in reverse osmosis, and this is what we call sodium potassium pump. So the energy for this transport is provided by hydrolysis of ATP. Hydrolysis of 1 ATP transports 3 Na plus ions out of the cell and 2 K plus ions into the cell. Kidney and brain uses 70% of the energy from ATP for this transport. The enzyme adenosine triphosphate that present on the cell membrane serves as Na plus or K plus pump, and it has internal and external sites for both Na plus and K plus. When the outer sides are filled with the K plus and the inner with the Na plus, a conformational change occurs. See, there is a conformational change occurs and the enzyme rotates. There is a special term for that. Keep in mind. So when the Na plus as well as K plus ions attached with the enzyme rotates, and the enzyme is present in the cell membrane. In animal, we don't have cell wall, we have a cell membrane. So this enzyme is present in cell membrane. So when Na plus and K plus ions are attached with the cell membrane or the enzyme rotates by 180 degree. So those who are inside the cell will be outside, and those who are outside the cell will be inside. Okay, and it is called eversion, that will be discussed in detail. This is a revolving door for transport of these ions in opposite direction. See, the eversion is actually a revolving door. Okay, the things sticked on the door, after it rotates, will be outside, and those who are sticked outside the door after rotation through 180 degree will be inside the cell. And this is the mechanism. See, this is a portion of the enzyme. This altogether there may be a cell, and we just cut a portion here, and this is altogether will be a cell, and we are just mentioning a piece of the cell membrane. So first of all, 3 Na plus binds with the enzyme or cell membrane. Okay, so this is inside the cell, and this is the outer portion of the cell. So I am just showing a piece of the cell membrane, and it is here, and this is inside the cell, and this is outside the cell. So first of all, 3 sodium ions binds with the cell membrane. So for binding, it requires energy, and it is ATP will provide energy for the binding. So bind 3 Na plus inside the cell. Then ATP also binds with the cell membrane, which is called a phosphorylation. The binding of ATP is called a phosphorylation. The ATP will provide energy for the binding of 3 Na plus. Then eversion takes place, rotation through 180 degree takes place, then those who are inside the cell will be outside. Then these 3 Na plus will be released into the extracellular fluids. Now still we can find the ATP molecule is here. Then from the extracellular fluids, 2 K plus ions binds. From the extracellular fluids, 2 K plus ions binds. 2 K plus ions binds, and it is here. Then dephosphorylation takes place, binding of K plus, as soon as the binding of K plus ion finished, the ATP molecule will detach from the cell membrane, and it is called dephosphorylation. Then again eversion takes place. Then the potassium ions in the extracellular fluid will be inside the cell. Then release of 2 potassium ions takes place. Then again the cell membrane will be free. The cycle repeats with utilizing ATP. And this is how a sodium potassium pump works. Okay. Now biochemistry of zinc, that will be discussed in next session. Thank you.